Mystery about bees. Although all honeybees are genetically identical, they quickly take on roles within the hive. Few become queens, while most become workers. Once a bee is a queen or worker, the change is irreversible, and they fulfill that role for life. Yet, that is not the case for the subdivisions among the workers. Workers start out their lives as nurses, spending their time in the hive taking care of the queen's eggs. Yet after 2 or 3 weeks, will transit to a new role as foragers, traveling to find sources of nectar and pollen to bring back after 2 or 3 weeks. So how do bees with the same genetic makeup can undergo such incredible transformations? The answer appears to be epigenetics, a biological process meaning roughly above the genome. Epigenetics refers to the chemical tags attached to genes that help regulate how those genes are expressed. Epigenic processes can turn a gene on or off, or simply make the gene more or less likely to be expressed. A simple way of looking at epigenetics is like this. While traditional genetics describes the way the DNA sequences in our genes are passed from one generation to the next, epigenetics describes passing on the way these genes are used. Take an MP3 player, for example. It will contain a lot of songs, which are analogous to genes. But you may play them by playlist, by artist, or by genre. This information, playlist, artist, or genre, determines which songs are played and in what order. And this is what epigenetics is to genetics. It is a set of processes that affects which genes are switched on or expressed. Many researchers have been studying epigenetics over the past few decades, and it is currently an area of intense research activity. We now know that a part of how epigenetics work is by adding and removing small chemical tags to DNA. You can think of these tags as post-it notes that highlight particular genes with information about whether they should be switched on or off. In fact, the chemical tag in question is called a methyl group, which can be added to DNA by enzymes called DNA methyltransferases, DNMTs, to modify one of the 4 bases or chemical letters A, C, T, and G that makes up the genetic code of our DNA. In most cases, more methylated Cs in the DNA of a gene results in the gene being switched off. Honeybees provide us with a good example of how this can work. Though the queen and workers in a hive are female and genetically identical, there are many differences between them, both physical and behavioral. The clue to how this comes about lies in royal jelly, a secretion that is fed to some developing larvae, which results to their becoming queens rather than workers. A fascinating piece of research showed that if the amount of the methyl group adding DNMT enzyme was artificially reduced in bee larvae, then the larvae developed into queens, even if they weren't fed royal jelly. This means that the switch between queens and workers can be flipped by the abundance of methyl tags on the bee's larvae's DNA. Fewer methyl tags lead to switching on of genes in the developing larvae, which result in the development of the larvae into queens and not workers. The researchers also discovered the role of epigenetics in the life course of the worker bees by carrying out a clever experiment. First, they looked to see whether there were epigenetic differences between groups of nurse bees and forager bees. They identified epigenetic differences in over 150 genes. The researchers then played what might be considered as a cruel trick by some. When the foragers were away, they removed all the nurse bees, an act which they knew would cause some of the foragers to revert to being nurses after they returned. When the researchers analyzed the bees' epigenetic tags again, they found that around 50 of the genes they had previously identified as being epigenetically different had changed back. This suggests that the changes to the epigenome were largely responsible for the changes that occurred when a bee changed roles.